Your midbrain develops from the mesencephalon brain vesicle. There we have seen the formation of vermis and also right and left cerebral hemispheres, then formation of tectum, tegmentum. What nucleus we have seen? What are all the 4 nucleus that develop in your midbrain region? Which nucleus develop in your midbrain region? Okay, I'll start with the forebrain in this session. If those topics are not completed, just let me know once, okay? So your forebrain develops from the which brain vesicle? It develops from your prosencephalon brain vesicles, okay? What are the parts of prosencephalon brain vesicles? It is telencephalon and diencephalon. Your telencephalon will give rise to which structure? Your telencephalon will give rise to hemispheres of your cerebrum. Cerebral hemisphere, okay? And your diencephalon will give rise to thalamus, epithalamus and hypothalamus structures, okay? So if you take the cross section of your prosencephalon brain vesicle, this is how the structure will look. Okay, this is your telencephalon part and this is your diencephalon part, okay? So during the development of your forebrain, we can see the differential growth in your telencephalon part and your diencephalon part. As a result, your telencephalon will grow rapidly and it will overlap your diencephalon part, okay? So once that changes occurs, differential growth. Okay, this is how the structure will appear. Okay, this is your overgrowth of telencephalon. And this is your diencephalon part now, okay? This is the diencephalon part. So when you see the adult structure, this is how exactly the cerebral hemispheres will appear. These are the 2 cerebral hemispheres, and here we can see the thalamus regions. Okay, this is how your adult structure will appear. Now we will see what are the changes that happens in your telencephalon and also in your diencephalon parts, okay? So whatever the structure you are seeing here, this is called as the groove of your telencephalon part, and this area is called the roof part. This is called as roof part, okay? And these structures what you are seeing here is your lateral wall of telencephalon, and these cavities will later grow into which ventricle? These cavities will grow into which ventricle? It will grow into your lateral ventricle. Okay. So now we'll see what happened in your groove part. I'm going to draw only the half structure. A part of your groove, this region, will start invaginating into your lateral ventricle part. So once the invagination takes place like this is how the structure will appear. Okay. So this structure is called as choroid fissure. This is called as choroid fissure. This choroid fissure happens because of the invagination of your groove region into your ventricle region. Okay. From the outer aspect, this choroid fissure is lined by your which matter? It is lined by your pia mater. Okay. So once your choroid fissure lined by your pia mater, it is called as telecardia. Okay. Now later in the process of development, your telecardia region is filled with the plexus of capillaries. Okay. Once the telecardia is filled with the stop talking. With the plexus of capillaries, it is called as choroid plexus. This structure is now called as choroid plexus. And your choroid plexus helps in the formation of your CSF fluid. Okay. If you remember the ventricular system and foramen, I have clearly mentioned CSF first or in the initial stage it will produce in your lateral ventricle region. From there it will flow or circulate into your central canal part, right? So this is how the formation of your CSF takes place in the choroid plexus region. Okay. Any doubts in this? Any doubts? Okay. So these are the total changes we can see in the groove of your telencephalon part. So what happens in your lateral walls region? Any idea? What happens in the lateral ventricle region? In your lateral walls, we have two parts. Okay. This is the first part and this is the second part. The first part is called as superficial thin part. Okay. And the second part is called as basal thick part. This is called as basal thick part. So where we can see the grey matter and white matter in your cerebrum? Your grey matter is present in which aspect? Inner aspect or outer aspect? Outer aspect, okay. Grey matter present in outer aspect and white matter is present in inner aspect. What is the reason? The neural cells present in your mantle cell zone or layer will migrate out of the marginal cell layer. As a result, in your brain region, mostly the grey matter is present in the outer aspect, whereas the white matter is present in the inner aspect. But during this process, not all the cells present in your mantle cell layer will come out or migrate into the outer part. Few cells will remain in that mantle cell zone only. So those cells which are remain in the mantle cell zone will form a structure in your basal thick region. This is called as corpus striatum. Okay, this structure is called as corpus striatum, and your corpus striatum basically have two structures or two differential parts like this. Okay, like this, and one like this. So now this structure is called as corpus striatum medially, and the another structure is called as corpus striatum laterally. Okay. Any doubts? So whatever the cells that do not migrate out of the mantle cell zone remains in your basal thick part and forms the corpus striatum. Your corpus striatum have two divisions. Those are corpus striatum medial and corpus striatum lateral parts. Okay. There is one more structure that pass between your corpus striatum region like this. Okay, this structure is called as internal capsule. This structure is called as internal capsule. Remember, your internal capsule cross or pass between your two strands of the corpus striatum region. We can see two nucleus in this region. Okay. The first one is caudate nucleus. The first one is caudate nucleus, and the second one is lentiform nucleus. Second one is lentiform nucleus. Your caudate nucleus is present deeper to your internal capsule, whereas your lentiform nucleus is present superficial to internal capsule. Okay. These are all the changes we can see in the groove and also on the walls of your Telencephalon brain vesicle. Any doubts? Any doubts in this? Okay. Then we'll see what are the changes that happens in your diencephalon part. Your diencephalon structure will appear something like this. Okay. And your diencephalon will show two sulcus, one like this and one like this. Okay. Because of these two sulcus, we can see three swelled areas in your diencephalon region. The two sulcus are basically the epithalamic sulcus, and the second one is hypothalamic. Stop talking or you both can leave the hall. Hypothalamic sulcus. Okay. So this is the epithalamic sulcus and this is the hypothalamic sulcus. Okay. The three swellings that present here are thalamus, thalamus, hypothalamus and epithalamus. Epithalamus and hypothalamus. Your thalamus is present between your two sulcus. That means your thalamus is present between the epithalamic sulcus and hypothalamic sulcus. So this is the thalamus region. Upper to the thalamus we can see the epithalamus part. Lower to the thalamus region we can see the hypothalamus part. Okay. These are all the changes that takes place in your diencephalon brain vesicle part. Any doubts? Any doubts in this? This is how the development of your forebrain takes place in the developing embryo. So how the development of forebrain takes place? What is the procedure? At first, your telencephalon will overlap your diencephalon part. Okay, next, once the overlapping of your telencephalon takes place, there will be a formation of groove, roof and lateral walls in your telencephalon brain vesicle part. There will be certain changes in both your groove and also in the lateral walls. Okay, so what are all the changes we can see in the groove? First, there will be invagination of your groove into your lateral ventricle part. So that invagination structure is called as choroid fissure. That choroid fissure is lined by your pia mater, then it is called as telecardia. Okay. Later the telecardia is filled with the plexus of your capillaries, and that structure is called as fluid. Okay. Then what are the changes we can see in the lateral walls? Your lateral wall is divided mainly into two parts: superficial thin part and basal thick part. In the basal thick part, there are few cells from your mantle cell layer that does not migrate and remains there only. It forms a new structure called corpus striatum. Your corpus striatum is again divided into two more parts or division, you can say. It is called as corpus striatum medial and corpus striatum lateral. Okay. Between these two structures, there is one more structure that runs. It is called as internal capsule. Deeper to the internal capsule, we can find the caudate nucleus, and superficial to the internal capsule, we can find the lentiform nucleus. Okay. These are the important nucleus that present in your cerebrum hemisphere part. Okay. So what are all the changes that takes place in the diencephalon? Your diencephalon will shows the two sulcus and three swellings. Two sulcus are named as hypothalamic sulcus and epithalamic sulcus, and three swellings are called as thalamus, epithalamus and hypothalamus. Epithalamus is present on the upper region of your thalamus and hypothalamus is present on the lower part of your thalamus. Remember these points, it will be helpful for your gross structures. So any doubts in this? Any doubts in the development of forebrain? Okay. Then we'll start with the development of your eye. So what are all the structures you can see in the development of eye? Or in general, what structures you see in your eye? Development of eye. So basically your eye is, development of eye will be studied under two main headings. The first one is development of optic vesicle. Okay, and the second part is development of various parts of eyeball. Okay, these are the two main headings to study the development of your eye. Okay, first we'll start with the development of optic vesicles. Okay, your optic vesicles are nothing but the outgrowth or out pouch structure of forebrain. Okay, your optic vesicle is nothing but the outgrowth or out pouch like structure present in your forebrain region. So this is your prosencephalon, mesencephalon, rhombencephalon and spinal cord part, right? So here in this region we can see some outgrowth which will later develop into, this is the outgrowth which will later develop into your future optic vesicle. Okay. So now if I take cross section of this area, I can see what is this structure? This is your neural plate. Okay, and this is your surface ectoderm. If you remember what we have discussed in the development of neural tube, these are the structures we have discussed. Okay, this is neural plate and this is surface ectoderm. So on either side of your neural plate, we can see a mass of cell present has a thick structure. Okay, during the development process, these thick structures will start invaginating towards your neural plate side like this. Okay, once the invagination takes place, this is how the structure will appear. Okay, now this invaginated structure is called as optic sulcus. Okay, this optic sulcus will further protrude in the downward direction like this to form your optic vesicle. Okay, so remember, first we have the neural plate. On either side of the neural plate there is a mass of cell which present has a thick structure. Okay, later this thick structure will start invaginating towards your neural plate cell, forming a optic sulcus. Later this optic sulcus will protrude more in the downward direction to form your optic vesicle. So this is your optic vesicle. This is how the formation of your optic vesicle takes place. So this is more like a cross-section study. If you see the same structure in your developing part, this is how exactly it will look. First it will appear only as a mass of cell like this. Okay. Later this mass of cell will turn into a sulcus like this. Okay. And later this optic sulcus will turn into your optic vesicle part. Okay. So this is your prosencephalon or your developing forebrain part, and this is the optic vesicle, and the structure that is connecting your optic vesicle into your prosencephalon, this is called as optic stalk. Okay, this is how the formation of your optic vesicle takes place. Any doubts? Any doubts in this? This is a narrow constricted area. Your optic tract is a narrow and constricted structure that connects your optic vesicle into your developing forebrain part. Any doubts in this? No doubts? Sure? Okay. So what are all the different or various parts we see in the eyeball? What parts or what structure we see in your eyeball region? What? What structures we see in the eyeballs? Histology is finished, right? So tell me at least the layers. What structures we can see in the eyeball? Okay. Development of the forebrain. The diencephalon develops from the median portion of the prosencephalon. Its cavity is called the third ventricle. The primitive diencephalon consists of two thick lateral walls, a thin roof, and also the floor plates. Each lateral wall represents a sulcus that is called the hypothalamic sulcus, which appears to be rostral communication of your sulcus limitans. The hypothalamic sulcus divides your lateral ventral into dorsal and ventral regions. Okay, that is nothing but your superficial thin part and basal thick part, what I have explained. The dorsal region develops into the thalamus. The ventral region enroaches on the floor plate and forms the hypothalamus region. The telencephalon consists of a median part and also two lateral diverticulum or the cerebral vesicles. The developing cerebral hemisphere enlarged forward, upward, and also in the backward direction. As the vesicle grows backward, it overlaps successively the diencephalon part, mesencephalon, and also the cerebellar rudiments. Though this thin wall, the choroid plexus of third ventricle protrudes laterally into lateral ventricle along a line known as choroid fissure. Corpus striatum develops bilaterally in the floor of the telencephalon adjacent to your thalamus part. Primitively, these areas of grey matter are sensory motor control centers. These points it will be discussed in your gross part in details, okay. Hence, these fibers that form internal capsule on each side divide the corpus striatum into two parts, that is a caudate nucleus, which can be seen in the dorsomedial portion, and lentiform nucleus, which can be seen in the ventrolateral portion. Okay. This is the structure what I have explained. You can follow the textbook pictures also. If you find it difficult, you can follow the diagrams which I have shown. Okay. Any doubts in the development of forebrain? Okay. For the clinical part, you have to write the defects of the neural tube only. NTDs, that is nothing but your anencephaly, rachis, and also your spina bifida, hydrocephalus. These things you can write for all forebrain, midbrain, and also for hindbrain, and also for your spinal cord. Okay. Then we have the topic development of eye. The development of eye begins on day 22nd after the gestation. At first, a pair of shallow groove or sulcus appear on either side of the forebrain. It is called as the optic groove or sulcus. The optic sulcus invaginates the surrounding mesenchyme to form the optic vesicle. Each optic vesicle grows laterally. Its distal part expands to form the optic vesicle. Its proximal part connect to the forebrain, constrict to form the optic stalk. Okay. As the optic vesicle comes in contact with the surface ectoderm, this is the formation of lens. So, any doubts in the formation or development of optic vesicle? No doubts? Okay. Take down that first point. Development of eye begins on day 22nd after the gestation. Okay. The different parts of or various parts of eyeball that we see here is lens. Then what are the other parts we see? Okay, sclera, cornea, next choroid, then optic nerve, important one, then ciliary bodies, iris. Stop talking. Last two rows, just leave the hall please. Yeah, next. cornea and retina, right? What else? Suspensory ligaments, okay, suspensory ligaments. Anything else? Okay. First we'll start with the development of your lens, okay? Development of lens. So this is your optic stalk followed by your optic vesicle. Okay, remember in front of this structure we have the surface ectoderm. This is your surface ectoderm, optic vesicle, and this is your optic stalk part. So during the development of your lens, your surface ectoderm will slowly start invaginating into your optic vesicle part. Okay. So once the invagination starts, this is how the structure will appear. Okay. Later, your surface ectoderm will completely invaginate into your optic vesicle part, like this. Okay. First, your surface ectoderm is present just in front of your optic vesicle. Now this region, which is present anterior to your optic vesicle, this is called as lens. This is called as lens placode. Okay. Once the invagination starts, there will be some changes in the structure of your optic vesicle and also in your surface ectoderm part. Now this region, this is called as lens pit. Okay. Once the complete invagination takes place, both your optic vesicle and your surface ectoderm structure will completely change. So now your optic vesicle is called as optic cup. Okay, and this surface ectoderm part is called as lens vesicle. This is called as lens vesicle. So by the time of 33rd day of gestation, whatever the surface ectoderm part that invaginated into your optic cup region will completely get separated from your surface ectoderm part. Okay, so now the structure will look exactly like this. Okay, this is your optic cup and this is your optic stalk, and this is your surface ectoderm. Only this part will get separated from the surface ectoderm and present here like this. Okay, now this is nothing but your lens. This is how the formation of your lens in the eye takes place in the 33rd day of the gestation. Any doubts? Any doubts in this? First we have the surface ectoderm present anterior to your optic vesicle. So this particular part is called as lens placode. After the invagination of your surface ectoderm or the lateral placode part into your optic vesicle, it is called as lens pit. Later, once the invagination complete, it is called as optic cup and also lens vesicle. At the 33rd day of gestation, your lens vesicle will completely get separated from the surface ectoderm. As a result, the right or up and down surface ectoderm will connect with each other once again and form the initial structure like this. Okay, and whatever the lens vesicles get separated from your surface ectoderm is going to form your future lens. This is how the formation of lens in a human takes place. Any doubts? Any doubts in this? No doubts? Okay. Comes in contact with the surface ectoderm, it induces it to thicken and forms a lens placode, that is the primordium of your future lens. The lens placode get depressed below the surface to form the lens pit. As the lens pit deepens, its edges approach each other and fuse to form a spherical lens vesicle, which is soon cut off from the surface ectoderm. As the lens vesicle is being formed, optic vesicle begins to invaginate to form the double layered optic cup. Remember, the optic cup which is formed here is the double layered structure. The margins of the optic cup grow and enclose the lens vesicle, that is the lens vesicle enters into the optic cup region. You can see the structure here. Compare the diagrams with what I have written. This one is for the formation of your optic vesicle and this one is for the formation of your lens part. Any doubts? Any doubts in this structure, in this figure? This diagram is to show the formation of your optic vesicle along with the optic stalk, which is in connection with your forebrain part. Any doubts in this? Subsequently, a fissure called choroidal fissure develops on the inferior aspect of the optic cup and the optic stalk. So I have already mentioned about the formation of choroid fissure in the development of your forebrain. Remember the points, okay? The vascular mesoderm enters into this choroid fissure and gets trapped in the optic vessel and also in the optic stalk region. The choroid fissure later gets closed. This vascular mesoderm, within it forms the hyaloid vessels, that is hyaloid artery and hyaloid vein, which supply to the layers of the optic cup, mesoderm of the optic cup and also to your developing lens vesicle. The distal part of the hyaloid vessels eventually degenerate, whereas their proximal part persists as central artery and also the vein of the retina. So what you have understood? How the formation of choroid fissure takes place, we have learnt in the forebrain. Remember, the choroid fissure is also present on the inferior aspect of your developing optic cup and optic stalk. So at one point during the development, your choroid fissure will get closed. So in the optic stalk region, later your hyaloid vessels will grow in that particular region, okay? In the hyaloid vessels, the proximal part will get degenerate, distal part will get degenerate, only the proximal part will be remaining. And that remaining part will give rise to your future vein of the retina and also the central artery, okay? This is the diagram, you can compare the points once. If any doubts, I will discuss. So this is your optic stalk region. In this region, we can see the choroid fissure, okay? Later, this choroid fissure will get closed. As a result, in the optic stalk region, don't concentrate on this region, just concentrate only on this region, okay? Your hyaloid vessels are growing here, okay? Later, this is the structure from above to below. We can see the outer layer, inner layer, hyoid artery and hyaloid vein part. Okay. Any doubts in this? Next, we have the development of various parts of the eyeball. The very first topic is lens. The lens developed from the lens vesicle, which is a derivative of your surface ectoderm. Initially, the lens vesicle is lined by a single layer of cuboidal cells. These points I'll discuss later. First, for these 2 points, see this structure. Whatever I have explained here, no, this is the same picture. Try to compare the structures once. First, we can see the lens placode followed by lens pit. Then we have lens vesicle. At last, we can see the formation of lens. Any doubts in this? Okay. Next, remember now to just try to imagine the circle part, that means only the lens part. Okay. So first, in the initial stage, your lens, this part is only lined by single layer of the cuboidal cells. Okay. The cells of anterior wall of the vesicle remains cuboidal, whereas the cells of posterior wall of the lens vesicle will get elongate, becomes columnar and extends into the cavity of your lens vesicle. So what you have understood? Okay. Now we have the lens. Okay. Imagine this is the lens and this is the anterior wall and this is the posterior wall and this part is your cavity. In the initial stage, your lens, stop talking. In the initial stage, your lens is lined by the cuboidal cells, single layer. Okay. After certain developments, only in the anterior wall, the cuboidal cells will remain. These are the cuboidal cells. But the cells present in your posterior wall in this region will now turn into the columnar cells like this. Okay. The posterior wall of the lens vesicle elongates and it will become or turn into columnar cells and extend into the cavity. This is the cavity part. It is getting extend into your cavity, and as a result the cavity of the lens is obliterated. So at one point your columnar cells will grow completely into your cavity region and your cavity will completely get closed. Okay. As a result, all the nucleus present in your columnar cells will be lost. So now the cells will turn into your columnar cells, or the posterior wall cells will be turned into fibers. Okay, which is avascular in condition. What do you mean by avascular? Yeah, right. So the posterior part of your lens is avascular. This part, this will be the avascular part. Only the anterior part of your lens is vascular. Okay. Any doubts? Any doubts in this? The cells in the anterior wall of the vesicle will remain the cuboidal, whereas the cells of the posterior wall of the lens vesicle elongate and it will also become columnar. Later it will extend into the cavity of your lens or lens vesicle. As a result, the cavity of lens is obliterated, that means the cavity will be closed. Okay. The elongated cells of posterior wall of the lens vesicle further elongate considerably and lose their nuclei to form highly transparent primary lens fiber. So whatever the lens fiber will form after losing their nuclei will be referred as primary cell fibers or lens fibers. The lens grows because new lens fibers, that is the secondary lens fibers, are added by cells in the equatorial zones of the cells. So which one is the equatorial zone in that picture? This one. This is a junction between your posterior wall cells and the anterior wall cells. Okay. This is where we can see the secondary lens fibers. Okay. These secondary lens fibers will help in the formation of new lens fibers. Okay. The cells of the anterior layer persist as epithelium. Although the secondary lens fibers continue to form during the childhood and lens increases in its diameter, the primary lens fibers become old and harder. At first, the lens is supplied by hyaloid artery. We have already seen how the formation of hyaloid artery takes place. So tell me how the formation of hyaloid artery takes place. How the formation of hyaloid artery takes place. In the optic stalk region, we have choroid fissure. At one point during the development, your choroid fissure will get closed and give rise to your hyaloid arteries. Okay. A branch of the ophthalmic artery. Later on, distal part of hyaloid artery will disappear and blood supply to the lens is stopped in this region. Consequently, the lens becomes an avascular structure. Remember, only the posterior wall part will become avascular, whereas the anterior part will remain vascular structure. Okay. Any doubts in this? Any doubts in the development of lens? Okay. This is the picture what I have trying to write here. Just try to compare the structures once. In the C picture, you can see your columnar cells losing their nuclei and turning into primary lens fibers. Okay. So this will be your equatorial region where the formation of new cell fibers or lens fiber, that means the secondary lens fibers, will take place. Okay. Any doubts? Any doubts in this? Okay. Next we'll see the development of retina. So what are the layers of retina? How many layers are present in retina? Nerve fiber layer, okay. Outer plexiform layer. Outer nuclear layer. Stop talking. Okay. Next. Okay, inner nuclear layer. Okay. Outer. Call your friend. Okay, ganglionic cell layer, inner plexiform, next Layers of rod and cones. Can anyone name all the 10 layers in your retina? At least seeing your books. Can anyone name all the 10 layers at least by seeing your books? You do? Say it louder, everyone should hear. Okay. Is it right? Others also say. So in the development part, we have two important layers. The first one is neural layer. Okay. In few textbook, it is mentioned as neurosensory layer. Okay. And the next one is pigmented layer. Pigmented layer or pigmented epithelium. Okay. In the development stage, these are the two important cell layers we concentrate in the retina. Okay. Your pigment layer is present in inner aspect or outer aspect? It is present in the outer aspect. Okay. And your neurosensory layer is present in the inner aspect. Okay. Now imagine this is your optic stalk followed by the okay. So now this is the outer pigment layer of your developing retina and this is the inner neural layer. Okay, and your entire optic vesicle structure is divided into two parts like this. Okay, this part is called as inner thick part and this part is called as outer thin part. Okay, and your neural layer will give rise to 9 different cell layers of retina. Okay, and your pigment layer is the one separate layer. This together makes the 10 layers of your retina. Okay, so in the development of retina, we can mainly see the two cell layers at the developing stage. The first one is pigment cell layer which is present in the outer aspect and the second one is neurosensory cell layer which is present in the inner aspect. The neurosensory layer will give rise to 9 different cell layers in the adult structure. Your pigment cell layer will develop into one cell layer in the adult structures. So which one is the pigment cell layer in your adult structure? Which one? Say it louder. Retinal pigmented epithelium, popularly known as RPE. Okay, this is all about the development of retina. Any doubts in this? We have the development of retina. The retina develops from the walls of the optic cup. Outer thin layer of the optic cup forms the pigment layer of the retina. Inner thick layer of the optic cup forms the neural layer or neurosensory layer of the retina. The cells of the neural layer proliferates to form various cells of retina, that is the photoreceptor cells like rods and cones, bipolar cell layer, ganglionic cell layers. The rods and cones are modified neurons present in our retina. What do you mean by modified neurons? Ganglionic cells. Okay, any other answers? But here we have already mentioned about the ganglionic cells. Again we are telling that rods and cones are modified. At least what is the significance of rods and cones in our retina? What is the significance of rods and cones in our retina? Helps in light and dark vision. Okay, any other answer? The axons of ganglionic cells form nerve fiber layer which passes through the optic stalk and forms the optic nerve. During embryonic and early fetal periods, the pigment and neural layers of retina are separated from each other by a space called intraretinal space. Intraretinal space, optic nerve, everything I will discuss in the next session, okay? Representing the original cavity of the optic cup. Before birth, the space between the inner and outer layers of the optic cup is obliterated with the proliferation of cells in the inner layer. As a result, the rod and cone cells come in contact with the pigment cell layer of your retina. Okay. Matrix cell layer will give rise to your rods and cones. Mantle cell zone will give rise to your bipolar cells, ganglionic cells, and also some supporting cells. Your matrix cell zones have the axons of the ganglionic cells. Okay, rods, RODS. It's a typing mistake. Okay. Any doubts in the development of retina? Okay. This is the structure what I have explained here. You can check it. The proximal most part of the outer layer, this part is going to form the epithelium of your ciliary bodies and also to the iris. Okay. Any doubts?